Hello everyone. Today we'll discuss the mechanism and process of fertilization. What do you mean by fertilization? It is a process of union of male pronuclei with the female pronuclei. In simple word, we can say it as a what? The union of male gamete with a female gamete or fusion of male gamete with a female gamete where the what the nuclei of both the gametes or pronuclei of the both the gametes fuse together to form the zygote. Both the gametes are haploid in nature and when they fuse together they form the diploid zygote. This is the electron microscopic picture of the ovum during the fertilization, and this just here is the what the entry of the sperm into the ovum, and this is the just after fertilization there is a formation of the second polar body during after the fertilization. This is the structure of the egg or the ova, and here the what the this is the metaphase 2, that is the second meiotic spindle, and this is the ooplasm. This is the first polar body, and it is covered by the vitelline membrane, and next to this there is a zona pellucida, and outer layer it is the follicular cells, that is the corona radiata. Then this is the structure of the human ova and the sperm, and that comprises of nucleus, then cytoplasm, or that we called it as ooplasm, then the vitelline membrane, and this is the corona radiata, and after that this is the zona pellucida layer, and the outermost layer or the outermost follicular cells. These are of the corona radiata. This is the human sperm having the head, their middle piece and the tail part, and this at the anterior position there is a acrosome, and these are the two things, one is a male gamete and another one is a female gamete. Both are required for the fertilization. The union of these female pronucleus with the female nucleus forms the diploid zygote, and the process we called it as a what? The fertilization. What are the steps during the fertilization? The sperm nucleus containing the haploid set of chromosome and acrosome containing the enzyme. These bind to the or the come near to the word the ovum, and these fuse with the word or it fuses to the word the these cells or the outer follicular cells of the corona radiata, and it releases the acrosomal content, that is a hyaluronidase enzyme, and it breaks down or regenerates the cell of this corona radiata. And after dissolving the corona radiata, it again dissolves the another layer, that is a what? The zona pellucida, and then it crosses the vitelline membrane, and after that the what? The sperm nucleus get entry within the ova. So these are the steps which are related with this. You can observe this is the meiotic spindle, and the sperms are trying to enter within the ova. This outermost layer is the corona radiata, then next to is the zona pellucida and the vitelline membrane. And as soon as the what this acrosomal content is released, what happens? This zona pellucida layer get dissolved along with the what the corona radiata, and the sperm gets entry towards this. This male pronucleus, this is a male pronucleus and the female pronucleus. what happens when there is an entry of the sperm into the ovum, there is a release of the second polar bond at the time of fertilization. And these fuse together, or the chromosomal content of these fuse together to form the zygote. And the structure which is formed after the fertilization is the zygote. This is the diagrammatic representation of the mechanism of fertilization in the sea urchin. In the sea urchin, or in the ovum of the sea urchin, instead of their corona radiata and the zona pellucida, the jelly coat membrane is there present on the vitelline membrane. This is a jelly coat membrane. This is a vitelline membrane, and above that there is a jelly coat. The sperm which comes near the what this ovum by the chemotactic movement or chemical attraction movement and acrosomal content, that is this is released, and this jelly coat is dissolved by this acrosomal content, and after that there is an entry of the this male pronucleus into the ovum or the ooplasm, and as soon as there is an entry of the this sperm nucleus into the ovum, what happens, these peripherally placed cortical granules rupture or break downs, and it forms a cortical granule membrane, or it forms the fertilization membrane. Then the acrosomal or the cortical reaction happens, and it forms this cortical membrane which avoids the polyspermy. This is the first step, that is a contact, and second is the what, contact and recognition, and second step is the what, acrosomal reaction, where the acrosomal content is released and the jelly coat is dissolved. Next to that, the growth of the acrosomal process, and due to that, there is an entry of the this nucleus or the male pronucleus. This fused with the or this male gamete, it get or this acrosomal process get fused with the plasma membrane, and then all its content it is released inside the ooplasm, and as soon as it gets entry, these cortical granules rupture and it forms a membrane, and that membrane is called as a fertilization membrane. The process is called as a cortical reaction. What are the steps? Contact and recognition, then acrosomal reaction, growth of acrosomal process, fusion of plasma membrane of the sperm and the egg, then the entry of the sperm nucleus, and last one is what? The cortical reaction, that is to form the fertilization membrane. The ovum, like any ordinary cell in its structure, and it is a large oval cell which varies from 117 to 142 micron in diameter in humans, and it having the coverings. It has a two membrane. Inner membrane, it is called as a vitelline membrane, and the outer thick transparent membrane, it is the zona pellucida. And next to that zona pellucida, there is a two or three layers of the cells which surrounds the zona pellucida, and that we called it as a corona radiata. Its cytoplasm, its cytoplasm is called ooplasm. It having the nucleus and the germinal vesicle. Its nucleolus is called as a germinal spot. The cytoplasm contains a nutritive material which nourishes the embryo in the early stages of development. Then the granulosa, this is the structure showing the granulosa cells, then the corona radiata cells, and these are the sperm which are reaching to the what ovum. And these are the cortical granules which are arranged at the peripheral position. Then these are the cortical reaction occurred and there is a formation of the fertilization membrane. This is the same picture or the steps that are involved in the fertilization in the sea urchin. First is the sperm approaches the egg, then the sperm acrosomal enzyme digests the egg jelly coat. The proteins on the sperm head binds to the receptor. Then the plasma membrane of the sperm and egg fuses together. The sperm nucleus enters the egg cytoplasm and this cortical reaction occurs and fertilization membrane forms. And after that, the sperm nucleus and the egg nucleus, they combine together or fuse together, and that is the process of fertilization, and after that there is the formation of the zygote. So these are the steps which are involved in the process of fertilization. Fertilization is a process by which the male and female gamete nuclei fuses together to produce a diploid zygote. There are the two types of fertilization. One is external and another one is internal. If eggs are liberated in water and fertilization occurs outside the female genital system, then it is an external one. The female lays a large number of eggs, then the male pour its sperm in the same region in water. The process and fertilizes the egg, then it is called as external. Example in fishes and amphibians. Internal fertilization occurs in animals that have a well-developed reproductive system. Animals may be oviparous or viviparous. The oviparous, where the zygote develops in a shell, that is the birds. And viviparous, the zygote develops inside the uterus, example the mammals. The intrauterine life is about 21 days in rat, 70 days in the guinea pig, while it is about 280 days in the human beings. The fertilization or the process of fertilization having the four major steps that we have earlier discussed. First is a contact and recognition between sperm and egg, that it recognizes their own species. The regulation of sperm entry into the egg, only one and inhibiting the others. Then the fusion of genetic material of the sperm and the egg and the activation of egg metabolism to start the development. Then the important process which is related to the sperm function during the fertilization is a sperm capacitation. What is the sperm capacitation? It is an enhanced sperm function. It results from secretion in the female's reproductive tract. Freshly ejaculated sperms are unable or poorly able to fertilize the ovum. Rather, they must first undergo a series of changes, that is, the molecular changes, and these changes or this process is called as a capacitation process. During capacitation, the cell membrane of the sperm may be altered by changing its lipid composition. Particular proteins or carbohydrates on the sperm surface are lost. Certain proteins are phosphorylated, in other meaning activated, that is, the proteins gets activated. The membrane potential of the sperm is dramatically lowered from -30 millivolt to the about -50 millivolt. Then increase rate of metabolism. The flagellum beats more rapidly. Re- the sperm becomes more motile. The changes in sperm plasma lemma proteins allow the sperm egg binding and occurrence of the acrosomal reaction. The proacrosine, which was inactive, is converted into the active acrosine. The sperm became capable of chemotaxis or recognize the chemical on the surface of the ova. Finally, the capacitation appears to destabilize the sperm's membrane to prepare it for acrosomal reaction. Only capacitated sperm can pass through the corona cell and undergo the acrosomal reaction. Then during the process of fertilization, the two main problems are there. How can sperms and egg meet in a dilute concentration in the external fertilization? And what mechanism prevent the sperm of one species from trying to fertilize the eggs of other species? The two mechanisms involved to solve these difficulties. The one is a species-specific attraction of sperm, and second one is a species-specific sperm activation. The sperm activation, that is a acrosomal reaction, is what it is a discharge of hydrolytic enzymes from a vesicle in the acrosome of the sperm cell. It occurs in two steps: the fusion between acrosomal vesicle and sperm plasma membrane, and second, the extension of the acrosomal process by polymerization of globular actin molecules into actin filaments, and initiated by soluble egg jelly or by contact with egg itself. And it can be activated artificially by increasing the calcium concentration. This is the acrosomal reaction. This is the acrosomal content which is filled in the acrosome of the sperm, and during that it gets rupture and it releases entire acrosomal content towards the outer covering of the ovum, either the jelly coat or the corona radiata. In the first, that is a contact and recognition, the plasma membrane of the sea urchin egg is surrounded by the vitelline membrane and the thicker outer covering that we have earlier discussed, that is a jelly coat. And in case of the mammals, it is a zona pellucida. Then during the acrosomal reaction, the acrosome of the sperm, it releases a proteolytic enzyme and acrosome digests the path through the external covering. The acrosomal process of sperm contact the vitelline envelope of the egg, and if sea urchin gametes are of the same species, a protein called binding on the acrosome adheres to the specific receptor on the vitelline membrane. In the step 2, that is a sperm and egg fuses together and the microvilli of the egg membrane form a fertilization cone. This is an electron microscopic picture of the fertilization cone. It forms a fertilization cone. Sperm is drawn into this cone and the gamete plasma membrane fuse and the sperm is drawn into the egg cell, so that by the fertilization cone, this the sperm nucleus is drawn into the what? The ovum, towards the ovum. The preventing the entry of the sperm more than the one sperm, that we called it as a preventing the polyspermy. Fast block to polyspermy involves egg plasma membrane action potential that prevents the additional sperm from entering, that is minus 70 micron to plus 10 micron, also releases the calcium. The slow block to polyspermy in mammals, the zona pellucida sperm receptors are modified to prevent the further entry of the sperm. In some organisms, They do not block the polyspermy. In salamanders, many sperm enter, but only one survives and rest are the degraded. And the third step, that is the activation of egg. Prior to fertilization, the egg is in quiescent stage or in arrested in metaphase stage of the second meiotic division. Upon binding of the sperm, egg rapidly undergoes a number of metabolic and physiological changes that are collectively called as a activation of the egg or egg activation. First, what happens during egg activation? The aerobic respiration increases in the egg. The enzyme system become activated. A burst of protein synthesis begins, and in most animals, the nucleus undergoes the second division of meiosis, and the meiosis gets completed afterwards. Then the step 4 is a pronuclear fusion, that is a fusion of male pronucleus and a female pronucleus. Once the second polar body is ejected, the female pronucleus can fuse with the male pronucleus. And this is the genetic beginning of the new organism. The haploid genetic complements of the two pronuclei form two and nucleus or the diploid nucleus, which prepares the nucleus and cell for the cleavage process. Then during the as soon as the sperm enters or the sperm pronuclei enters into the ova, there occurs one reaction, that is a cortical reaction. These cortical granules are present peripherally on the egg. The fusion of egg and the sperm membrane stimulates a series of changes in egg cortex and known as a cortical reaction. These chemical reactions change the egg's cortical granule. Granules fuse with plasma membrane, releasing enzymes separating the vitelline membrane from the plasma membrane. Swelling lifts the vitelline membrane, and thus there is a formation of the membrane that we called it as a fertilization membrane. And once the fertilization membrane is formed, it prevents the penetration of other sperms and prevents the polysperm. Then when the chemotaxis or the first process occurs, that is the this recognition and the contact, the important process occurs, and that is called as a chemotaxis. What is that chemotaxis? It is a chemical attraction that operate in the lower organisms where the fertilization in the external. And that process it is called as a fertilizing anti-fertilizing reaction. Where the fertilizing is present and where the anti-fertilizing, the jelly covering of the egg had a hypothetical receptor molecule, and these egg which having the receptor molecule, these are called as a fertilizing. And the fertilizing molecule have receptor sites which are complementary to the surface molecule, and that we called it as a anti-fertilizing, which is present on the sperm head. Means on the surface of the egg there is a presence of the fertilizing, and on the surface of the sperm head there is a anti-fertilizing. And the fertilizing and the anti-fertilizing, they can recognize each other, their own species. The fertilizing molecules in the sperms at receptor site and cause the clumping. The modern view holds that fertilizing and anti-fertilizing are analogous to lock and key mechanism. During agglutination, the fertilizing keys fit into the fertilizing locks. The anti-fertilizing keys, it fits into the fertilizing, that is the locks which are present on the surface of the egg. Fertilization has high specificity and will only agglutinate the sperms of the particular species, and it is thought to be due to definite molecular arrangement of the amino acid and monosaccharide in the fertilizing molecule of the species. This is the structure of the agglutination or the mechanism of agglutination, and that is a fertilizing anti-fertilizing reaction. The particle of the fertilizing, it is present on the ovum surface, and the anti-fertilizing or the respective anti-fertilizing, it is present over the surface of the sperm, and thus they the fertilizing and the anti-fertilizing of their own species, they recognize each other and they agglutinate to complete the process of fertilization during the recognition and the contact process. These are the diagrams of the process of or mechanism of fertilization, how the process of fertilization occurs in the organisms. That is, the first is a reaching the sperm and the acrosome towards the egg envelope or toward the egg surface. Then in the second step, these are the same steps, the approach of the sperm towards the egg. Then second is the opening of the sperm tip after its contact with the egg membrane. This is the egg membrane and this is a sperm tip which attached to this. The acrosome and the sperm plasma membrane becomes continuous. In the C diagram, the acrosomal wall is everted, and in the D, the sperm and the egg plasma membrane interdigitate and these both get fused. And in the E diagram, the fertilization cone is formed. And lastly, in F, the sperm that gets entry towards the ooplasm or toward the egg cytoplasm to for the fusion process. So these are the steps which are related to the process of the what? The fertilization. Thus, these are the processes just we have discussed. That is, the first is a penetration of the corona radiata, during which the acrosin and trypsin released from the sperm acrosome, and the due to that it dissolves the corona radiata and the zona pellucida layer. Then the penetration of the zona pellucida. The zona is a glycoprotein shell that maintains the sperm binding mediated by ZP3 protein that induces the acrosome reaction. It releases the acrosomal enzyme, penetration of the zona pellucida, and contact with oocyte plasma membrane. And after that, permeability of the zona pellucida changes on contact, the cortical reaction occurs. The fusion of oocyte and the sperm cell membrane, and after that, both head and the tail of the sperm, it enters the cytoplasm of the oocyte, but the sperm plasma membrane, it remains behind. Actually, there is a fusion between the pronucleus of the male and the pronucleus of the female, and to form the zygote. Lastly, what are the effects or consequences of fertilization? Fertilization stimulates the secondary oocyte to complete the meiosis II to form the second polar body. It restores the normal diploid number of the chromosomes, that is 23 chromosomes from the male and 23 from the female. They fuses together to restore the diploid number, that is a 2n number that equal to the 46. During the process of fertilization, it brings about the recombination, which is a cause of variation of human species as maternal and paternal chromosomes intermingle. The embryo contains only maternal mitochondria because the sperm mitochondria are dispersed into the egg cytoplasm and discarded. It determines the sex of the embryo, and Initiation of the cleavage occurs just after the fertilization. Without fertilization, oocyte degenerate into the 24 hours. So these are the consequences or effects of the fertilization. Thank you.